Good morning, I'm Vic Senese, and today we have breaking news from the front lines of the fight against antibiotic resistance. Researchers at MIT have harnessed the power of generative AI, artificial intelligence, to design brand new antibiotics, compounds that could take on some of the most dangerous drug-resistant bacteria threatening public health today. Published in Cell Today, the study describes how the team created antibiotics capable of combating two tough opponents, drug-resistant Neisseria gonorrhea, which also causes gonorrhea, and multiple drug-resistant Staphylococcus aureus, better known as MRSA, MRSA. Using advanced generative AI algorithms, the researchers designed and screened over 36 million possible compounds, many of them completely new to science. The best candidates they found are structurally different from any antibiotic currently in use and seem to work in novel ways by disrupting bacterial cell membranes. For decades, the development of new antibiotics has slowed to a crawl, and those of us who've been around for a while know that. In the last 45 years, only a few dozen have been approved, and most were just variations of other existing drugs. Meanwhile, our friend the bacteria have been evolving, rendering many of these drugs ineffective. Globally, drug-resistant infections are estimated to cause nearly 5 million deaths every year. This AI-driven approach opens the door to exploring chemical spaces we've never been able to reach before, designing completely new molecules rather than relying on what's in existing chemical libraries. Now, the MIT team used two main strategies. First, they gave their algorithms a promising chemical fragment known to have antibacterial properties and asked them to generate millions of possible molecules around it. One compound, called NG1, emerged as a standout. It was not only effective in lab tests, but also cleared drug-resistant gonorrhea infections in mice. Those mice. Second, they tried an unconstrained approach, letting the AI freely create molecules within the rules of chemistry. This time, they targeted MRSA. That search yielded DN1, which successfully treated MRSA skin infections in mice. Both NG1 and DN1 appear to work by targeting bacterial membranes, but in different ways. And because they are structurally unlike existing antibiotics, they may bypass the current resistant mechanisms. A nonprofit partner, Fair Bio, is now working with MIT to refine these drugs and move them towards preclinical testing. Researchers also plan to use the same generative AI tools to find treatments for other dangerous bacteria, including Mycobacterium tuberculosis and Pseudomonas aeruginosa. Pseudomonas A. In the words of Senior Professor James Collins, this work shows the power of AI from a drug design standpoint and could represent a turning point in the global battle against superbugs. If successful, this approach might not just give us a few new antibiotics, it could open a floodgate to entirely new classes of life-saving drugs. Imagine a world where your doctor uses artificial intelligence to spot problems before you even feel symptoms, where surgeries are guided with superhuman precision, and the rural patients get the same expert care as those in major hospitals. That's not science fiction. It's happening right now, and it's happening in urology. For ongoing updates, keep it right here. I'm Vic Senese. Thanks for watching UroNurse News.
From bladder to prostate, we've got your back. UroNurse News, we're on the right track.